second reproducibility talk this year. I've just started recording. So today our speaker is Emma Ganley. Just a little bit about Emma. Emma is a director of strategic initiatives at Protocols. She formerly worked in scientific publishing for about 15 years, and she used to be chief editor of PLOS Biology, and she then rejoined PLOS Biology in 2011 and been with them for quite a few years. So Emma followed a standard research career path. She obtained a PhD from the MRC-LMB in Cambridge, UK, and then she had a postdoc in US, and then she relocated back to the UK and worked at the University of Dundee. She's now with Protocols, and she's been passionate about all things related to open research, open data, open methods, reproducibility, and scientific integrity. So without much ado, I will request Emma to start the talk. You can leave your questions in the chat function, and we will be sharing the recording afterwards.
That's great. Thank you. Thank you, Shafag. And yes, thanks for inviting me to talk today. And I apologize if anyone tried to join the talk that was previously scheduled in October that we had to unfortunately delay until now due to weather. I'll say British weather, we're not very good at coping. So I'm up in Scotland, and we were hit hard in October by the storm that passed by. But yes, thanks, Shafag, for the introduction. That's right. My kind of background is in research and then in publishing. And now I am at protocols.io. So I've been with Protocols since the start of 2020. And what I'll talk today about is around how you can achieve reproducibility by sharing your research methods on protocols.io. So there's a kind of general background that I'll run through as well about why it's actually important to share research methods. So regardless of platform, if you don't even use protocols.io or you don't think it would be so suited to your research, it's still very important to consider sharing your methods alongside anything else that you're putting out with your research. So data, analyses, etc., etc. It's very important to share the methods too, and I'll try and persuade you of that. I will give an introduction though to protocols.io, which is the tool platform that I'm representing, and we'll show you a little bit what that looks like, and if internet works well, I will give a little bit of a demo, but there should be lots of time for Q&A, so if you have questions, please do pop them in. So I'll get started with that general background. So something that perhaps some researchers will be aware of because they will have experienced it, but others may not have quite clocked onto, is that often when you try to search the research literature for previous methods, you can't find them. The information is missing. So here's just a few examples of tweets from, as was known, Twitter, now X. On the left-hand side is a biologist. Sorry, that GIF gets annoying after a while. The researcher was looking for a protocol in a published 1997 paper, where it's noted that it was as described in another paper in 96. In that paper, described as in another paper in 87, and eventually maybe that 1987 paper was paywalled. Perhaps it's actually only available in a paper version, and you won't be able to access it if your university doesn't have it or your institution doesn't have it in their library, or you're not affiliated and you don't have access. So you can't find that information. That is not only true for biologists. Here's a physicist who had the same issue when looking to see how a device was created, was made. Devices were fabricated as previously described, and the original reference has devices were fabricated with conventional methods. So what does that mean? We have no idea. We don't know how these devices were actually fabricated. So this is an issue for reproducibility and repeatability, that researchers can't find access or get to the information, like experimental or other methodological details that are important for the context of that research, or if you want to try and build upon that research and so on. There was an initiative carried out by the Center for Open Science in the US with Science Exchange called the Cancer Biology Reproducibility Project. So this initiative wanted to try and replicate the experiments from 50 published cancer research papers. And what this showed is that when looking in the papers, the methods details weren't there in the same way as illustrated by those tweets, but actually also when talking to the authors of those studies, the information wasn't available. So going back to the researchers themselves was not fruitful either in attempts to replicate and have sufficient information to repeat the experiments. There's a really great write-up about this in The Atlantic by researcher Ed Yong. I'll paraphrase here, and he interviewed the researchers who attempted to carry this out. So he said the hardest part by far was figuring out exactly what the original labs actually did. So papers should have methods that ought to provide recipes for doing the same experiments, but often those recipes are incomplete, missing out important steps, details, or ingredients, and in some cases the recipes aren't described at all, and researchers simply cite an earlier study. So repeating and building upon previously published work is hard. In fact, they halted this study after just 18 attempting the experiments from 18 of those published papers. And when they went through, when attempting to prepare replications of 193 experiments from 53 papers, there are a number of challenges. 0% of the protocols were completely described. But they, as I noted, they weren't able to get the information from the researchers. So there's a bigger issue here about than just what's in the manuscripts that end up being published. It's about how researchers are keeping that content and making it something that can be drawn upon in the future if somebody would like that information. But also for themselves, if they wanted to repeat the experiments they'd performed previously, they actually don't have enough information remaining in the lab. Maybe postdocs moved on, maybe it was just in a lab notebook that's now can't be found, etc., etc. They don't have that information to repeat it themselves either. What do we care about the methods? This is an image taken from a magazine, actually just baking magazine, of some cookies that were baked just by making tiny tweaks to one single recipe. So making a tiny tweak to the method resulted in something phenotypically very different. You can see that the cookies are all very different from one another. So if the cookies are your data or your results of an experiment, analysis of size, thickness, texture, hardness, flavor, anything that you actually would observe about those cookies, you can only analyze and really interpret what they mean if you know the tweaks that were made to the method and the changes that were made to the method that was used to create them. So those tiny little tweaks, a tiny bit more flour or a bit more sugar or using the wrong ingredient, can change massively the results that you get from your experiment. And we do really well at sharing data now. So a lot of effort's gone into making sure that researchers share data alongside research papers. We have data availability statements now alongside many, many research papers. But if we've got all of these data, how reproducible are they? Actually, they'll only be as good as the methods that have been shared alongside them. If you want to reanalyze or try to replicate or reproduce, you really will need to have the method as well. So there's a lovely quote from an evolutionary biologist, Dobzhansky here, that nothing in biology makes sense except in the light of evolution. So I switched that. I have my own take. Nothing in research data makes sense except in the light of how it was generated. So you need to know, you need the methods to be able to reuse and reproduce data, to make value in these masses and oceans of data that we're now keeping to make available to the research community. It's critical to get the methods alongside them. Okay, so I will show you protocols.io, which is a tool that can be used by any researcher to share the methods as a part of their research process. we have a very simple mission. We want to make it easy to share method details before, during, and after publication. So all the way through the research process. Oh, I meant to update the numbers on here of the total public and private, and I forgot to do that from the after since October. I did it last time. So it's perhaps known by many as an open access repository. So there's a good page here, just protocols.io/welcome, which is a good place to go to if just want to have a look and a browse at content that's been posted on there. So anyone who comes and actually publishes a protocol on protocols.io, it will be available with a CC BY license, and you can come and just have a look at them on there. They are FAIR as well. So if people know that FAIR acronym, they're findable, accessible, interoperable, and reproducible. So you can use to kind of come in and poke around this open access repository. When you place content on protocols.io, it's very much a bare template, so you can use it across all different research disciplines. It was originally created to accommodate life sciences, biomedical sciences, purely on the background of the one of the co-founders, Lenny Teitelman, who's a geneticist, but it has now been used across all different research disciplines. If you have any kind of step-by-step approach that you would like to include comprehensive details of in the context of a research study, you can put it onto protocols.io. It's collaborative, so you can share with colleagues in exactly the same way you would any kind of Google document or Office document, where you can share selectively with individuals, with a group of people, and you can control the permissions that each user has, whether they can edit or just view, etc. So you can collaboratively create the protocols with colleagues. And I've mentioned that you can really use this throughout the research workflow. So I've got just a vague schema to try and represent. If we start over here on the top right, a researcher would be defining a question that they want to investigate, start thinking about outlines and plans and exactly how they want to probe a research question. We would come around to planning some experiments and methods here, maybe identifying some protocols that are already available out there or something that someone else has been using in the lab. Think about the equipment and materials, sourcing all of that information and putting it somewhere together. So at this point, you can already be using protocols.io. In fact, there may be protocols on there that are useful to you in the context of the question that you want to start investigating. So you can have protocols on protocols.io, you can share and collaboratively edit those with your colleagues. And you can also seek and obtain feedback via comments. So if you want to start planning these, but get feedback from your group leader, they can have access and can give you just comments on them high level rather than editing specifically. So when we're happy we've got a good plan and a good approach, we might start actually running the methods. So protocols.io is brilliant here. You can run the protocols dynamically on the platform, and for every run that you make, you can add notes. If you make a change to the protocol that day or something strange happens, you can make a note of all of that, but you can record every run. So you can have a date time-stamped record of the run, which will correlate with any data that you generate on that day. So we can do this here. We can have the run record, analyze results, assess the results. Was it successful? And then we get to this next bit where maybe it wasn't successful. Perhaps we need to tweak the approach and rerun and reanalyze and continue. And we probably end up looping here a little bit if we're developing the method and optimizing and trying to make sure we have a replicable experiment that is solid and rigorous, etc., etc., etc. Hopefully at some point we exit the loop and we have success, and we've got successful experiments. We can finish performing all of the research that we want, analyze the data, etc., etc., start writing up the paper. The brilliant thing here is that you can, if you've got that full protocol on protocols.io, at that point it's a trivial few steps, few buttons to publish the protocol, post it, if you will, like a preprint, post it and obtain a DOI for your research manuscripts, materials and methods. So you can have that whole detailed with all of the information, incredibly comprehensive description of the protocols that you followed, have the DOI and put that into a research paper. You don't really even need to then write the materials and methods section other than to say we performed X experiment, please find all the details in this protocol posted on protocols.io. So that's exactly what a lot of people now do. Of course, if you wait to the time of publication, it's a bit then long to add it at the end stage. But if you've been using it throughout the research workflow, it makes it a lot easier. So here's a PLOS biology paper where they have methods and protocols for blah, blah, blah. All of the different experiments are available as a collection on protocols.io. And so they have the link to the protocol collection. So if you follow the link, it will bring you to protocols.io. And what's really clever is that on protocols.io, the experimental process, we think of it as a dynamic thing, which should be possible to version, and continually evolve. Maybe you have additional information. Maybe you made a mistake actually when you published in there, or something needed clarifying. You can make new versions on protocols.io and post them really, really easily. This link will always take you to the version that was published when the paper was published, but you can also come to the newer version and you'll be able to compare and see what was updated from one version to the next. So we call this dynamic permanence because you'll always link to the static version that was published with the paper, but the authors can come and make changes. Perhaps they've got new equipment and they've further honed the experiment with a new piece of kit, and then you'll have a different version for different piece of equipment, etc., etc. We have formed a partnership a couple of years ago with PLOS ONE, who created this article type, which is called Lab Protocol. These are specifically interlinked with protocols.io so that the methods has to be published or posted on protocols.io. And there's very prominent, see the protocol, the materials and method will point across, and it's bidirectionally linked. And you can see on protocols.io that it's peer reviewed. And just to illustrate that we are way more than a life sciences platform or biomedical or just some disciplines. The very first paper that was published under this article type was an archaeological imaging methodology. So you can see it's very adaptable across different research disciplines. Okay, and what are some of the other benefits of posting the method separately? It means it can be posted with a different author list. So if the first author is somebody, maybe graduate student who spent ages developing this method but didn't get around to actually applying it to some novel, interesting research question, the method can be posted separately from the paper, and it can have a completely different author list. So you can actually get credit for the method as a separate standalone output. And when it's available as a standalone output, this is an example of how it can kind of form different connections between researchers. So on the top left is a researcher in Chile who was looking for RNA extraction, so a specific kind of experiment from primary cortical neuron cultures, so from a kind of brain cell. And there were a few tweets, and here there was a tweet from a researcher at UCSF, I think a postdoc at UCSF, and she pointed to a protocol on protocols.io, RNA extraction protocol. So it's a completely standalone research output. When we looked at that protocol, it had been published as a part of this paper in GigaScience, which was an analysis of 3-spine stickleback parasites, so a fish parasite. So it seems really unlikely that researchers planning to investigate neuron cultures would have happened upon this paper on a fish parasite as a likely location for a method that would have been valuable to them. But because the protocol was separately available as a standalone output, they found that it was identifiable as something that would be relevant to them because it was a separate output. So you can increase discoverability, obviously reproducibility, make these amazing research connections, enable reuse and enhance value of research. It's really kind of multifaceted in the ways that it kind of accelerates science, if you will. So key benefits: manage and share research data and protocols. I've really shown it here, but simplify teamwork and improve collaboration. So you can have these workspaces where you can be working with all of your colleagues, collaboratively editing. Also just know where everything is, right? Have a central place to keep the methods, to keep the processes that are being developed by any research group. That's invaluable, and to be able to find things as people move on, as staff changes, as just people start working on different things, that overall should save time and help you keep work organized. Okay, so I will go on to show you a little bit how you can play around with protocols.io and maybe demo a little bit too. So I've got a couple of slides. First of all, just to say that if you're not interested in posting your own content, you may want to come and just have a look at what's on there. So from that welcome page, it's easy to start a search. So you can just pop in any kind of term that interests you, the research kind you like, or look for researchers with a key interest, and just browse through the results. You'll find, I think it's now a drop down actually, but I've not updated this slide. Publications or people or workspaces. So you'll find different methods that have been published. You can see some of the metrics that pertain to a given protocol. So, you know, how many times it's been looked at or downloaded, if it's been bookmarked, and other kind of key metrics that you can see to look for protocols that may be useful for you. Someone else's protocol, if you know that as they've written it, you can use it, you can just run it if you want yourself. You can also create a fork of the protocol. So what that means is you make your own copy of it, essentially, but we call it a fork because the lineage is preserved. So you can always see if someone's made a fork of a protocol, where it was forked from. So think of it a bit like a phylogenetic tree, I guess. And then that version, your fork, you can make modifications to it if you want, keep it private for yourself, or if you make substantial changes to it, you can even publish it, and that lineage will be visible. So within the context of FAIR, the reuse is more than ticked in terms of being able to make available things to other people, the research outputs. When you're searching as well, you can look for workspaces. So if you work in a particular subject area, you might search for public workspaces that are relevant to you, and you can have a look at the content. You can have a look at the researchers who are in there. You can actually seed discussions if you've got questions, or post comments or ask questions of the people who've been publicly posting protocols. And so one such example, public workspace, it's been very, the numbers here are much larger now than here. I haven't updated this slide for a while, but it organically grew. We made this space available for anyone, and this method development community all around coronavirus included content for sequencing, viral load, public health matters, all sorts of different things. So you can really see how sharing this way, kind of for public health response, can actually be very powerful at disseminating information in a speedy way across researchers globally. If you haven't and you want to, you can sign up for an account from the welcome page. The business model is such that anyone can make a free account and will have 2 private protocols, but you can always publish for free. So you can publish and post as many as you like. When you've posted one, you'll get another private one. You'll always be able to maintain 2 private files. It's very much a similar model to GitHub, however, where if you want more private space to develop things privately, then there's a pay model. And our goal really is institutional licenses. So UEA doesn't have one, but we'd be very happy to chat to people about that, or talk more about that. But there's a place to check if institutions have licenses. So I'm going to jump into Mo and show you briefly, but just to say, there's a file manager where you can store all your files, exactly what you would expect a file manager to be. There's a place to view and edit documents and protocols, which is a specialized format so that you can capture all the protocol information. And then there's a workspace profile page, which is that front top page, a bit like that coronavirus one I just showed you. So there's a few different reviews just to try and hopefully you won't get lost. So let me jump out of the slides and show you where we are. So here I'm in the file manager essentially. So this is my Emma's workspace. Every workspace will always have a top folder. Anyone who I invite to this workspace will be able to see all of the content in here. But if I want to keep something private, there's another folder that's just got my name on it. I can always keep content private in there. Then you can see you can put whatever file structure you want. You just create new folders, dump files. You can actually drag and drop. So if you've got a ton of PDFs on your laptop scattered here, there and everywhere, and you'd like to centralize them somewhere, you could just put them all into a workspace so you knew they were somewhere where they'd be preserved and kept all together. If you belong to more than one workspace, they'll all appear down the left-hand side. So I have a lot. Most researchers wouldn't have that many, but you can imagine maybe you would have your own workspace, one for your whole team or a specific collaboration or project. So you can organize content in the workspaces as well. And let me show you what a protocol looks like. So on the kind of reproducibility theme, this is one of my favorites that I use for workshops often. So I've made this one, making a British cup of tea. If you think about a method that is in a research paper, normally it's a couple of paragraphs, perhaps of just narrative text, which is not very useful necessarily for explaining exactly how something was done. What you have with protocols.io, you can add disclaimer if it's public health or there's something else you want to disclaim, have an abstract for it because it needs to be independently findable. But otherwise, you have this lovely step-by-step format. You can have sections. So we've got the different kind of heating the water, brewing the tea, adding milk. You can create your own sections. You can also add images and short videos. So you can really illustrate and make it very, very clear, set up what setup should look like, what a specific procedure should look like, the kind of any kind of step that you would follow if you're greeting people in a cohort and you want to always be consistent with the way that you greet them. Anything like this, if you have a script or if you have a computational workflow pipeline, you can put this all in a step-by-step way. If you have got times, you can use components in there. And then if I'm running the protocol, I can actually run a timer dynamically when I actually run through the protocol. So if there's a step of waiting or just letting something run for a period, you can do that way. So I can run a protocol very easily here. If I click, it will take me, I can add a record, run the protocol, we'll go next. It lets me step through everything. So that part is done. Can track the progress at the top, done. If I make a change somewhere, I can either edit the actual step, which doesn't edit the whole protocol, it will only edit the step in this run. I can add a comment to it, and I can do things this way. So I won't do that now. Here's the timer, so I can set my timer to run for 3 minutes, so I can make sure my tea is brewed to perfection. And go on this way, etc., etc. And when I'm done, I can save the run. So now I can save this as done, performed today. We won't put it in the coronavirus methods, we'll put it in my workspace, and we'll pop it in run records and say save. And if I jump back to that file manager now and refresh, and I'll go into the, where did I pop that? In the run records. There'll be my run record from today is stored in here. So I have this, I can keep it. And if I'm working with an electronic lab notebook, or I want to put with other files somewhere, or with the data that I generate, I can export the files that I have, all the protocols as well, to many different places. So we have a link in with Lab Archives on electronic lab notebook. We do have integrations with another couple, RSpace and SciNote. And from those electronic lab notebooks, you're logged into the notebook and you pull from protocols.io, but it verifies into your account. So there's lots of different things that we can do here. And let's see, if you have a protocol that you're editing, so this then is the editor view, so you can add sections, We can add another section, or we can add steps. And you just type down the left as a toolbar that's kind of relating to the steps and the parts of the protocol that way. Along the top is your standard kind of editing toolbar. You can pop links in, or videos and images, tables, other things like this. And these are what I call the components. So if you have specific variables that are relevant, like amount or a sample or duration, you can pop these in here. You can add in citations using kind of links from DOIs, and they will pull into us under the reference tab if you add citations. There's also smart component, which is kind of key value pairs. So if you want the run record to require someone to add some specific observations that are happening during the protocol, it basically makes them put in the value of the observation, whatever it might be, when you actually make the run record. So you can kind of enforce obtaining specific information. So actually that could even be used for questions and answers to a degree. There's lots of other things I won't go into too much. The sections give you the table of contents, which is visible here. But also if you're viewing the protocol, so like that T1 I showed earlier, you can also see the table of contents from the top left, and you can navigate through using those as well. So if it's a long protocol, it lets you jump in around in the protocol. Actually, I can show that better perhaps on this published one. So I mentioned that coronavirus methods development community earlier. Here's a protocol that was published very, very early on in the pandemic, January 22nd, 2020. So this predates most lockdowns and action being taken. Not much was known probably generally around the world at this point in time about coronavirus. However, these researchers who work on pandemic preparedness and public health outbreaks, etc., were able to put this up very, very quickly. The reason they could put this quite long and complicated protocol up so quickly back then is because it was quite similar to one they'd already posted on Ebola, so on Ebola virus sequencing protocol that was actually posted in late 2019. So this is the forking that I mentioned earlier, where if a protocol is already available, you know you have a slightly different variation on it, you can make this fork and adapt it for whatever's relevant in that different context. So here it's just a sequencing protocol for viruses applied to different viruses with different processes. I mentioned you can compare. So if you click on more, you can do compare. I think I preloaded it earlier to save having to generate it on the fly. It's quite quick, but internet doesn't always work very well while I'm presenting. But here you can see that coronavirus is on the left, the Ebola virus sequencing protocol is on the right. So for example, one thing immediately that was different was the amount of template RNA being used, so the overall quantities and volumes. And they're highlighted red and green. And through the protocol, you'll see when there are things that are slightly different. So you can actually go in and have a look at what was updated from one protocol to the other, which is very helpful. And if you make a new version of any protocol, you can compare any version to any version. So we could, if we wanted, switch to different forks. I can see all the private ones because I have super user, but there is also multiple versions now of this protocol. So this is where the authors have come in and updated for whatever reason, perhaps to clarify, perhaps to add more information on the protocol. So it's been updated many times. It's for what concerns reproducibility. We also have this works for me button. So anyone who manages to use the protocol and have it working for them can come along if they're logged in and click the works for me button. So I won't click it now, but you can, if you click on the number, see who the researchers are that said they've got this protocol working. So you can check it's not just all members of Josh's lab, which it definitely isn't in this instance. But here's how that table of contents, those different sections become useful, because you can jump down. So you can imagine as well, sometimes if it's a multi-day process, you can have day 1, day 2, day 3, whatever makes sense depending on the nature of the research that you're working on. I don't know why that's showing up like that at the top. I think it's just a relic from here at the top. There are all these other places. When people make forks of a protocol, they can be shown up here, forks and versions. So in this instance, there's a lot of different versions that have happened. If we open all, you can see that other researchers have come along and made their own forks of this. So in terms of tracking reuse, you have a really great way of seeing how many times the protocol's been adapted by someone else and reused and published and so on in different versions. So this is an extreme example. Most protocols wouldn't have as much reuse as this, but it really illustrates just the value of making content available that way. You can also see all of the metrics. So for this protocol, well over 200,000 views of all the different versions, the forks, the comments. I didn't really show you the comments. Let's have a quick jump back. So when a protocol is being developed privately with you and colleagues, or when it's published, you can come along and put comments on individual steps. So, oops, if it's oh, that should be opening out for me. Let's just go to the comments. We can see there are a lot of step level comments where someone's come along on an individual step and posted a question, and the author has come back and put answers in. So you can see if I can get it to open now. Don't know why that's not loading. You should be able to see the comments that have been posted on the protocol. Let's go to the newer version and see if that's any better. So that's how it tells you, I told you earlier, that you can make new versions. So on this one, no, there's some bug happening here. I have to report this after this is the issue with live demos. It's not liking that at all. The comments and questions and answers, normally that works really well as like a public FAQ, where you'd come and have a look, and if you had a question about this step, possibly someone else has already asked it and had it answered. So you can have a look at the comments and the responses from the authors, and it may be that there's already an answer to your question if you have one. Can put all of the materials in here too. And I think that's most of the things to say on here. Oh, and yeah, metrics will also track when a paper's been cited somewhere, when a protocol's been cited somewhere, and we'll pull those references in if we spot the citations. And whenever a protocol's published, it gets this DOI. And so when we say published, it's not had peer review, obviously, it's been posted by the authors, more like a preprint. That DOI can go into a paper that would then be peer reviewed. And any different version will have a different DOI, so that they will always be linked to one another. You'll know that they're different versions of the same protocol. And they're added to different workspaces. So if you've had one for your collaboration or your research group has a workspace, you can clearly note that it's a part of that workspace on here, that you don't have to if you don't want to. Okay, I will pop back to the slides. There's just a couple more slides, I think, and then I can take the questions if you have any. So I showed you earlier how you can put that DOI into a journal publication, into your manuscript. There's well over 500 journals that recommend authors do this. If you actually look at the author instructions, they will be actively encouraging it. It's just, I think people don't often necessarily look at all of the author instructions. funders, it's becoming more and more common for funders to not only ask for data management plans, but also to know how methods and protocols will be available, how they'll be shared and stored, etc. So using a tool like protocols.io can be very beneficial. But if you're not using protocols.io, something versionable and digital is definitely the recommended way to do this. And more institutions are taking out licenses. So there's quite a few in the UK, Edinburgh, Glasgow, Manchester, I'm thinking off the top of my head, but there's a few and it's growing all the time. So if you think there might be interest at UEA, let us know. We also have protocol import service. If you have got protocols as a Google Doc, a Word doc, a PDF that you use all the time and you would like to get them on protocols.io, but you're not sure where to start or you don't have time, we do have a service. It does have a fee, but it's not huge, but it can be a really good way of starting to use the platform, because we have editors, all of whom have research backgrounds, who will do that import. It's quite a manual process, but they know all of the features of the platform, so can show really how you would present the information the best way possible on the platform. And you would have an account, and when you're happy with it, would get transferred back to you. And I think it just can really help researchers to see how their content can be shown and presented on the platform. We are quite a small team, albeit we have recently been acquired by Springer Nature. They are not changing particularly anything to do with our business model, but we now have a lot more resource and support to hopefully make further improvements to the platform and actually to work much more closely with many well-known research journals. So on that, many benefits to using a tool like protocols.io. I think I've gone through all of them: collaboration, discoverability, reproducibility, and reuse. That credit and recognition is definitely not to be underappreciated as a part of this. That's important for especially early career researchers, people who are actually doing the hard work of making the methods. Mentioned this dynamic permanence, the benefits of being able to version the methods, improving those materials and methods, but also stewardship of research output. So within a group, but also at a higher level across an organization, university. And so with that, I can take questions if there are any. There's a couple of links here. We often do other webinars, or people can sign up for demos. If they actually want to start using, feel free to sign up for a demo. There's a few UK time slots, and that would generally be me who would be giving them anyway, so people would know already who it would be. And I will stop the share and see if you've got any questions.
Thanks very much, Emma. This is so insightful, and it's like an entire new universe to me as a social science researcher. Yeah, obviously I'm curious if I could use it for my own research at some point. There is a question in the in the chat function. So Mina is asking if an embargo can be applied on protocols.
Okay, so not an embargo per se, but if you publish, we also have a way to reserve a DOI and generate a private link so that you can share it selectively with reviewers and editors if you've got it linked with a research paper, and only toggle finally publish when you're ready to. So if the associated paper's been accepted and you want to then make it go live at the same time, that's within your power to do so. So it's probably better than an embargo in a way, because it's completely under your control.
Right, thank you. I think you mentioned the version control possibility option. So I just wanted to double check if my understanding is correct. So if a protocol is updated, can previous versions be still accessed?
Yes, yeah. So you can always look at any one of those versions and compare and see what's changed, unless someone says to us, we need to take that one down. If maybe there's an image that's under copyright that shouldn't have been included or something like that, we can retract something. But generally, no, we want the transparency of all the versioning. I think, subscribe to the philosophy that all of research is an evolving process, and that it's helpful to be able to see that all the way through and see what changed and what evolved and what happened along the way in an open way. So yes.
Okay. And regarding the collaboration between researchers, so does the platform facilitate real-time communication among researchers?
Yes, so you can more than one researcher can be in a protocol and editing it at exactly the same time. So that's simultaneous editing in the same way you would a Google document. When you've got multiple in, you can see the different avatars at the top for who's in and editing. You can see who the users are who are accessing a protocol. You can see what changes individuals have made to it. And so it works very much like in that way.
Okay. And you did mention that it's not a peer-reviewed process, but I just wanted to clarify, so regarding the accuracy of the protocols. So is there any process to ensure that they're accurate?
So we're not reviewing. We screen to make sure no nonsense gets posted, because spam people will come along and try and post utter nonsense, or just telling kind of advertorials for CBD gummies and random things like this. In terms of will the protocol work, we don't test that. We're not checking that. We're not reviewing that. Over time, what you will see is that the metrics or the works for me button on the protocols that are used and are reused and are found to be valuable, they'll float to the top if you actually search on metrics, because they're the ones that are being verified and found. And if they're linked with a research paper, people will find them and use the good ones. So I think it's this kind of self-selecting thing of science, really, or research in general, that the good content will be findable.
For someone who is just starting to work with protocols, what kind of challenges they should be expecting, and how they can overcome them?
I think the biggest challenge, honestly, is probably the mental block of how to do this on a daily basis. I think it's that activation energy. So just starting to use the platform, and in a way, probably starting by playing around, just using it to do something like a cooking recipe is probably the easiest way to familiarize yourself and realize it's very intuitive. So rather than waiting for something that, you might get very frustrated if it's your research, just coming in and having a play around with the tool, and signing up, asking for help. We're very, very responsive. We are only a small team, but respond very quickly to requests for support or help, or if something's not working, I will report the bug about the comments, and probably it'll be fixed later today. So just let us know if something's not doing what you want it to, basically. I think it looked like Rose had her hand up.
Okay, yeah, Rose, please.
Hi, Emma. Thanks very much. That was really nice. I would apologise if I ask you something you did cover, because I had a lab member come running in the office and grab me, and I had to run out to the lab to deal with something. So I'm sorry. I just wanted to start by saying that I thought that this was something that's been needed for a really long time. At least for us, I find that the word count in papers actually prohibits us putting in fully detailed methods. Actually, it's just it's impossible actually to even share it properly. So this is really nice way of doing it. I wondered if you're looking for a protocol, do you have to have an account, or can you just look, or do you need an account just to search for?
No, you don't need an account to search and access to anything that's been published. They're all CC BY, so they're all completely available for anyone. So if a prompt comes and says make an account, you can probably just tell it to go away and browse through any of that publicly available content.
Cool.
If you want to comment or run, then you will need an account, because at that point you're interacting more with the platform. But the account is free to set up as well, so it's not, you're not signing up for anything. You can switch off emails if you don't want emails, but if you find it's useful and would want to advocate at UEA, we have had some contact there, but we haven't had success yet. But we could kind of try and work with you to advocate for a license that would mean you weren't limited on the accesses at that point.
I think the interest was probably more than perhaps it looks like in this talk. I will say that I had more than one link to get to this one, and the first link I tried didn't get me. It's the second link that I received that actually got me into this meeting. So I don't know if people disappeared into another, had a different link or something. But that aside, I just wondered, do all journals accept this as a reference in their materials and methods, or
we've never ever heard of a journal saying you must not do that ever.
Okay.
So
okay.
Even if it's not explicitly in there, we've never had a journal say don't.
Okay,
I think
cool. So it works.
It's a recognised way of sharing that this is a research output. Everything is going to be available through it, so everything's
right. Oh, that's really handy, because I'll admit I actually haven't seen it before, so it was a little bit new to me. And I just had one more little question, if that's okay. And I was just wondering about whether or not there are any issues with putting a protocol on there that might have a significant overlap with, say, the protocol within a, I don't know, manufacturer's kit of some sort of assay, and maybe you've made one tweak or two tweaks. Are there any problems with that? Or
so methods are funny in terms of copyright. They're not subject. They're actually not subject to copyright in the same way other content is. So if there's a step-by-step method available on anyone else's site, but you present it differently, this is kind of a nice, good to know information. If you present it differently in a different format, you're not breaking any copyright. Obviously, ideally, you'd be making some changes. But if you attribute and say, based upon the kit from X and we've made these adaptations, then I think it's just being transparent. But there are some vendors like NEB have got loads of their things on protocols.io. So you might even find that a vendor has some of the content on the platform already, and you would be able to make a fork of it and adapt for yourself too. So
that makes sense.
Depends who it's from and what it is, but that people do that, so they'll be taking an NEB protocol and saying this is our version of it applied in this specific situation.
Brilliant. Okay, that. You've just answered my follow-up question to that too. So awesome, thank you.
No problem.
Thanks, Rose. There is a follow-up question from Mina regarding the embargo issue. So if someone wanted to timestamp their protocol, is the only way by publishing the protocol? She's wondering if researchers would be worried about potential scooping without an embargo.
I mean, I'm not the best person to ask about scooping because I don't really believe in scooping. I'm a believer that when people independently do similar things, it's confirmation. So I'm also a believer in sharing early and transparently to accelerate science. That said, the timestamp for some people is obviously important. I don't know if publishing will get you the timestamp. The versions will have recent timestamps and history available with them. I think it's fallacy that scooping is a big issue, to be honest. Have you got evidence that someone is being scooped when they share a method before they publish the paper? I've not actually heard of any instances of it happening, I suppose.
Okay, thank you. I think Mina is typing a follow-up note. So yes, thank you, Mina. And on future developments, how does protocols IO plan to evolve in the future? Maybe new functions?
New functions, we're always taking feedback from researchers. So if we're in a different kind of expanding into a different research discipline and they suggest to us add these components, they're not in there right now, there are things you could make, it would be usable by a lot of researchers, then we try and add them. We have been making changes to the run functionality recently so that it can be split between more than one researcher, for example, or picked up and stopped and started a bit more. We do have some kind of scheduling task manager tools in there, and we'll be working on those. But we're also working on more maybe integrations with other electronic lab notebook providers, but there are many. And more integrations with journals, actually, as we've landed at Springer Nature. there are obviously a lot of journals there who we're really looking forward to working with, and implementing more integrations with some of those too.
Yeah, Rose, please.
I just suddenly thought, but and you may well have said this already. Like I say, I'm sorry if I missed it. But in our lab, we're a little bit. Well, we have issues with space, and taking things like laptops, etc., into lab spaces isn't really very practical a lot of the time. So I noticed in your in the protocols, it was very beautifully written out, but that's not what I can use in a lab to refer to as I'm doing something. So often I'll often want to print something out onto a bit of paper that I can scribble on as I go, that I will then go back. And so is there a printable version?
Yes.
Is there like a
Yeah, no, and some people are not allowed to take devices in the lab.
Yeah, exactly.
Point blank. So you can print it as a PDF and scribble all over it to your heart's content, and then write up notes wherever makes sense. So even you could even then go back to the desk and make the run and add the notes afterwards if you put them on paper, to keep that electronic version if you wanted to do it that way. But I mean, obviously everyone will use it differently, but yes, you can export PDF. You can just export the materials and materials if you want to kind of assemble everything first, and then have all the steps as a separate part to go through later on. So yes.
Yeah. Right, that works. So if you can export it as a PDF, I've just bought myself a Remarkable. I don't know if you've seen them, but I really love it, because you can import PDFs and Word files, etc., onto, and then scribble on them with your digital pen, and then kind of recreate that back out. And I think that would be it. Yeah, so it works. Yeah, thank you.
Yeah, of course. No.
Okay, thank you. Any more questions?
No.
Right. Please feel free to follow up afterwards if anyone has any questions by email, if they watch the recording back, whatever. Just emma@protocols.io.
Okay.
Thank you.
Thank you. We're going to share the recording on social media, so yeah, I'm pretty sure there will be more interest. Thanks very much, Emma.
Thanks, Shafag. Thank you.
Thank you.
Thank you.
Bye.
Thanks, everyone. Bye-bye.